Good morning, students. In our previous videos, we discussed about different floral parts and its function. Now, the male reproductive part, which is called as a stamen, and the stamen is made up of anther and filament. The actual reproductive part of a stamen is anther. Today, in this video, we are going to discuss about internal structure of anther. Now here in front of you a diagram showing parts of a stamen and three-dimensional cut section of an anther. The anther, male reproductive part of a flower. It is a bilobed, tetragonal, oblong, knob-like fertile part of stamen, and the lower part of anther is connected with a tube-like structure which is called as filament. The filament connects the anther to the receptacle in the flower. Now in front of you here is a tetrasporangiate anther, TS of tetrasporangiate anther. You can clearly see this normally anther consists of two lobes and each with two elongated microsporangia or pollen sac. So you can say that dithecous anther is a tetrasporangiate and anther lobes are fused by the connective tissue. In center you can clearly see the connective tissue. Actually anther is made up of several layers of cells, and these cells are highly specialized cells and perform different functions. They are responsible to carry out reproductive and non-reproductive function. The non-reproductive tissues includes the epidermis, endothelium, tapetum, circular cell cluster, connective, stromium, and vascular bundles. Each of these tissues are carried out a specialized function. Cells of these tissues perform a specialized function. In addition to these, anther also contain microspores, haploid microspores that fill the pollen sac and differentiate into pollen grains. Now here three figures are showing different portions of a typical anther. Figure A is showing the shape of the anther. There are four microsporangiums are present in each corner of anther. Figure B is showing a small portion of figure A. In this, four anther walls are clearly visible. Outermost is the epidermis. Just beneath the epidermis there is a layer known as endothelium. Beneath the endothelium there is a middle layer which is made up of 1, 2, 3 layers, and the last innermost layer is a tapetum which surrounds the microspore mother cells and provide them nourishment or nutrition. Figure C is showing mature anther after dehiscence. Here mature pollen grains are scattered inside and the anther wall is ruptured at the point called stromium. Here is a quick review of important functions of different cells and tissues. You can clearly see this: connective joint anther, and connect anther to filament and provide it a structural and structure and support. Next one, circular cell cluster, it helps in dehiscence. Endothelium, it provides a structure and support and helps in dehiscence. Epidermis, it is the outermost layer and it provides a structure and support, prevent water loss, gaseous exchange, and helps in dehiscence. Next one, microspore, pollen grain, development of pollen grain are important. It plays an important role in development of pollen grain. Next one is stromium, it helps in dehiscence, and very important layer is tapetum, which provides nourishment to the developing microspores, and it secretes enzyme for microspore release from tetrad. It also secretes several enzymes and helps in formation of exine. Next one is vascular bundle. Connection between anther and filament and flower, and nutrition. It provides a nutrition and water supply to anther. In the very beginning, the anther first appears as a cylindrical structure composed of a mass of meristematic cell, which consists of 3 germ layers designated as layer 1, layer 2, layer 3, which give rise to different anther tissues. You can see here layer 1 gives rise epidermis and stromium. Stromium actually it helps in dehiscence and greatly stretched and inflated in mature anther. It is located between the 2 locules of each anther lobe. Layer 2 gives rise archisporial cell, megaspore mother cell, endothelium and middle layers. Actually, archisporial cells are hypodermal in origin and consist of more vertical rows of cells along with dense cytoplasm and deeply stained nuclei. The cell divide periclinally and in form primary parietal cell towards the periphery and sporogenous cell towards the inside of parietal cell. Now the parietal cell undergoes a series of periclinal and anticlinal division to form 2 to 5 concentric layers of anther wall. Here endothelium, middle layer and 2 to 3 layer and outer layer of septum collectively form anther wall. Now the third germ layer give rise connective vascular bundle and circular cell cluster adjacent to stromium. Now this is a flow chart showing different germ layers, layer 1, layer 2 and layer 3, and different type of cells developed through these germ layers. One thing is very clear here. You can easily observe this. Layer 2 and layer 3 both are involved in formation of septum. The outer septum formed through layer 1, germ layer 2, and inner wall of septum is formed by germ layer 3. In this figure, you can clearly see development of anther with pollen sac. In figure A, you can clearly see anther primordia, which is differentiating in archisporial cell, epidermis, and further differentiation of archisporial cell into parietal cell and sporogenous cells. Further differentiate and form different layers: epidermis, parietal cell and sporogenous cell. So in last or in fully mature anther, there is a 4-layered epidermis. Just beneath the epidermis, endothelium. Beneath the endothelium there is a middle layer, 1 to 3 layers of middle layer, and next one is tapetum. Inside all these layers there is a microspore mother cell, and which develop pollen grains or microspore. In the last figure, as you can see, archisporial cell differentiate into microsporangia and surrounding tapetum and endothelial tissues, and simultaneously in each corner of the anther primordium, while vascular bundle differentiate within the center of the anther primordia and establish connection with the filament. Now you can clearly see different layers and its name: epidermis, endothelium, middle layers 2 to 3 layered, and tapetum outer and inner. constitute the anther wall. The outermost layer is epidermis. It is generally single layered and protective in nature. It divides anticlinally and try to keep space with the enlarging internal tissues of the anther. It helps in considerable stretching in the surface area. It provides the structural integrity to the anther and assisted in gaseous diffusion, prevent moisture loss, and it also helps in descent of the anther lobes. Just beneath the epidermis, there is a layer known as endothelium. It attains the maximum development before the descent of the anther. It contains fibrous bands, and due to presence of fibrous thickening, the endothelium is also known as fibrous layer. The endothelium is associated with a high proportion of alpha cellulose and a small amount of lignin at maturity. The specialized nature of the endothelium together with the stromium, it helps in descent of the anther. The third layer of anther wall is known as middle layer. It presents just beneath the endothelium. It is made up of 1 to 3 layers. The cells of middle layers degenerate to provide nourishment to the growing microspore mothers. The innermost layer is tapetum. It is the innermost layer of anther wall and usually derived from parietal layer. The tapetum surrounds the sporogenous tissue and attains maximum development when microspores are in tetrad stage, and finally when microspore convert or turn into pollen grain, all these cells degenerate. The tapetal cells enlarge rapidly and become filled with dense protoplasmic content as well as nutrients. Actually, the main function of tapetum is to provide nourishment to developing microspores and pollen grains. They are become multinucleated or their nucleus become polyploidy. Tapetum is of two types. One is amoeboid type and the second one is secretory type. The amoeboid type of tapetal cells fuse to form periplasmodium. It passes in between the sporogenous cells to nourish them, and whereas secretory tapetum pass out substances over the sporogenous tissues for their growth and differentiation. As you can see here, function of tapetum. So prime function of tapetum is to provide nourishment. Next one, it helps in formation of sporopollenin as well as pollen kit formed with the help of tapetum or after the secretion of tapetum. Amoeboid tapetum secretes enzyme like calase. So finally we can say tapetum is absorptive, nutritive and secretory in function. In this video we have discussed about internal structure of anther in detail, and in our next video we'll discuss about microsporogenesis. Thank you. Have a nice day.